6, 5, 4.
Hi, and welcome to Botany 101. I'm Angela Nishimoto. Okay, recall last time that we did evolution part 1. Okay, we ended with Darwin and some of his history, some of his ideas. And now we're going to do evolution part 2. Okay, we're going to go into the evidence for evolution. We're going to talk about microevolution and macroevolution. And finally, what is important in plants is polyploidy. Okay? So some evidence for evolution includes homologies. Okay, homologies are characters that are shared by different organisms. Okay, different organisms, different species. Okay? So in the mustard family, the plant family, the mustard family, or the Brassicaceae, and here are some members: broccoli, cabbage, radish, and stalks. All of them have a characteristic in that they produce juice when they're squeezed that smells like sulfur. Okay, that's actually a homology. That's a character that's shared by different organisms that are grouped together, like in this case in the mustard family. Okay, but similarities are not always from common ancestry like they are in the Brassicaceae. Okay, you have two groups: the African spurges, or the Euphorbiaceae, and American cacti in the Cactaceae. Okay, they tend to have similar succulent stems, or stems that hold a lot of water, because they both evolved in dry habitats, okay, to which they become adapted to. So these are plants that have different ancestry, and they've adapted in similar ways, okay, to similar environments, but in different places. Okay, the spurges in Africa and the cacti in America, or in the Americas. Okay, so they're not closely related. Okay, so this is an example of something called convergent evolution, where you have organisms evolving to form similar seeming structures, okay, even though they're not closely related to each other. Just like the ironwood trees. Okay, ironwood trees, people tend to think that they're pines, but they are not. Okay, these are flowering plants. That's an example of convergent evolution, where you have a plant that looks like it makes pine cones, but it's actually a flowering plant that makes fruits. Okay. Now, further evidence for evolution is the structure and relationships of proteins, DNA, and other molecules, okay, between different kinds of organisms. So in all organisms, okay, DNA codes for RNA, which codes for proteins. Okay. Another universal is the use of ATP, adenosine triphosphate. Okay, it is used as an energy source, a chemical energy source in all cells, all living things. Another universal is cytochrome C oxidase, an enzyme. Okay, so let's see, the use of ATP and also cytochrome C oxidase must have both evolved very early, okay, in life. Okay, so both of them appeared very early in the history of life on Earth. Okay, and they were then inherited by all organisms that are alive now. So that actually points to the relationship of all living things. Okay. Also, fossils tend to be simpler in the oldest layers and more complex in the younger layers. So you have simpler fossils that are older, more complex, and younger, so you have increasing complexity in many cases. And also biogeography is another piece of evidence for evolution, which is a geographical distribution of organisms. We talked about this earlier. Remember, we talked about Vavilov, right? So generally you have some groups that live only on one continent or island. So we have in the Hawaii, we have the silver-sword lions, okay? So this is a group of plants, okay, that first actually came from California and Mexico by one or very few founders coming to the islands, being carried by a bird, probably sucked in tar on the bird's feathers or on its feet, Okay, so this is a group of plants that actually diversified and adaptively radiated into environments. Yeah, but they only occur in Hawaii. Okay, so especially true of islands. Okay, so you have groups that occur only in one continent or only in one island group, like the Silver Sword Alliance occurs only in Hawaii. Okay, anyway, so now we're going to go look at some fossils over here at this display table. Okay, so here we are. All right, so some of these fossils are mollusks. Okay, mollusks have a soft body and a shell of some kind. Okay, so it's ammonite, this brachiopod, this gastropod, all these fossils, this pelecypod, these are all different kinds of mollusks. Okay, we have a crinoid worm segment here. Okay, we have a bryozoan, right? A coral or a piece of coral skeleton. Okay, we have dinosaur bone, and sometimes dinosaur bones, you could actually take a look at them. You can see the osteocytes or the cells inside them. Okay, here we have a shark's tooth, another vertebrate animal, a chondrichthyes or cartilaginous fish. Okay, and here we have a coprolite. This is actually fossilized dung of turtle, sea turtle, an extinct sea turtle. And here we have a little trilobite. So trilobites, so some of these fossils go back a long way, like tens of millions of years. Like trilobites actually go back to the Cambrian, to 570 million years ago. Okay, I actually used to have a trilobite fossil about yay big, about so big. Okay, but it got broken. You know, my cat knocked it off the file cabinet and it broke. I got it from northern Nevada, where they have lots of fossils in the desert there. So we have all these different kind of fossils derived from animals, and here we have the one lone plant. Okay, sometimes what happens is, this is some petrified wood. So sometimes what happens is that since we tend to be animals ourselves, we tend to focus more on the animals when we're talking about biology, right? And also there's also a little bias toward the terrestrial environment. Okay, because we live on land, we tend to focus more on terrestrial plants than on plants of the sea or of the water. Okay, that's our own kind of bias as animals who live on land, terrestrial animals. Okay, anyway, let's go back to the computer. So here we are again. Okay, back to the outline. Okay, now in terms of microevolution, that's generally evolution within species. Okay, so Darwin had seen that through artificial selection, animal breeders changed domestic animals, such as dogs, such as pigeons, such as chickens, such as cattle, such as horses, such as pigs, a whole bunch of things, okay, through animal breeding. Okay, we also talked about crop breeding. So he actually, when he set out his different theories for natural selection, he actually had certain observations and an inference. Okay, for one thing, you have overproduction. Okay, all living things overproduce. Okay, such as there might be one tree that makes thousands of seeds. Okay, only maybe one or very few actually germinate and grow to become another tree. Okay, mushroom too can actually make 1 million spores. Every one of these spores capable of producing another mushroom or another fungus. Okay. Secondly, you have a struggle for existence. Okay, in plants, all seeds or spores or other propagules, they need to compete for water, for light, for nutrients, for space. So of course, many die. So there's a struggle for existence. There's competition and struggle for existence between individuals. Okay. Thirdly, you have inheritance and accumulation of favorable variations. Okay. So all organisms vary, right? If you look around yourself, if you happen to be in among a crowd of people, you look at the people around you, all the people look different, right? So all organisms vary. All of us have variations within us, okay? So those that tend to be favorable or make us more fit for survival and reproduction tend to persist, And we tend to pass on these traits through sexual reproduction to our offspring. So as humans, we do that. Plants do that too. Okay? Or if you have asexual reproduction, so you have a plant that reproduces asexually, whatever made the parent plant successful will tend to make the offspring plant successful as well. Okay? Now, variations that are not favorable for survival tend to be lost. Okay? Because sometimes the organisms that have these traits that do not actually actively lend itself towards survival, they might tend to die before they reproduce. Okay? Then we have survival and reproduction of the fittest. Okay? So the best organisms that are adapted to each environment have the best chance to survive and reproduce. Okay? So like a tree that loses its leaves during the dry season, okay, and it can survive that and then flower, like the vili vili, will tend to be able to outcompete or actually do better than a plant of the same species that doesn't lose its leaves. And because it doesn't lose its leaves, it does more transpiration, it loses more water, so it might have less of a chance of survival, okay, than the vili vili that does lose its leaves. Okay? Now, mutations are changes in DNA sequence. It's the basis of all genetic variety within and between species. Okay, so mutations are changes in DNA. Okay, and the simplest of them is called the point mutation. Okay, which is just a change in one nucleotide pair. Okay, so this is actually the smallest mutation that could code for a slightly different protein. Okay, or it could code for more or less protein depending on what kind of gene, whether a regulatory gene or just a gene that codes for a protein is actually affected. Okay, we have deletions where a part of a chromosome breaks off and is lost. That's another mutation. Okay, we talked before about translocation where a part of a chromosome breaks off and is attached to another chromosome. And we also talked about inversions before, where part of a chromosome breaks and reattaches onto the same chromosome in an inverted way. Okay? See here on this illustration, you see a deletion here of a part of a chromosome. Okay, they also show you some different things like duplications. You can actually have duplications where one part of the chromosome duplicates itself and adds itself onto repeating areas. Okay? Here's an inversion where a chromosome within a chromosome or a gene or a series of genes within a chromosome actually inverts. It actually may curl in among itself, break off, and it is reattached in an inverted way. Okay? Insertions can happen where you have one chromosome. Here's chromosome 4. This chromosome 20. This is probably from a human karyotype. Okay, so a piece off of chromosome 4 becomes attached to chromosome 20. That's an insertion. Okay, and we can have a translocation. That's the same thing, where you have one gene from one chromosome actually moving to a different chromosome. That's called a translocation. Okay, so of course chromosome numbers can actually affect changes too. We'll get into that in a little bit. Okay. Now we also have another impact on evolution within species is migration. So migration is when you have gene flow between populations, when you have individuals moving from one population to another, or just exchanging gametes, or actually moving gametes from one population to another. Okay? Then we have the effect of something called genetic drift. Okay? Genetic drift is actually changes that happen to the genetic makeup of a population due to random events. And these come in 3 types. Okay? We have the founder effect. Okay, so where you have a home population, like say the tarweeds of California and Mexico. Okay? And you have only one or a few types that actually migrate to different areas. That's called the founder effect, where you have only one or a few. So it actually changes the population makeup of the places where the founders go to. Okay, so a good local example of the founder effect is the Hawaiian Silver Sword Alliance. Okay, and we have random chance. Okay, so random chance is just say for example you have some vili vili trees. Okay, in a parking lot, some people might have sowed some seeds and are growing these really trees in a parking lot. Okay, and then they're hit by some kind of an insect pest that spreads a fungus and starts to kill them. Okay, so one of the things you might try to do is try to actually either apply an inoculant, a pesticide to the trees to try to kill the pests and keep them from killing the trees, and a fungicide. Okay, or what they might do is they might actually just cut down the trees. That actually happened. There was a really tree in the Bishop Museum parking lot that actually had kind of like a light greenish colored seed. It's just very unusual, okay? But the next time we went there, I think once I found a couple of seeds under one of the trees there, you know, and one of the students took it home because I guess she wanted to someday make a lei from that. But of course, the next time I went back to the Bishop Museum the next year, they had cut the trees down because they had been infested with a pest. Okay? So random chance, just you have some genes or some alleles taken out of the population by random chance. They just happen to be there and they just happen to get cut down. Okay? Then we have the bottleneck effect. Okay, so what you have is you have a population at a certain size, and then you have some kind of a bottlenecking event where the population declines. You have some kind of event such as climate change, which is like a big bottlenecking event that happened several times in the Earth history. You could have a meteorite hitting Earth, making a big splash and creating a cloud, killing a lot of plants, that kills a lot of animals because animals need plants to live. And so you have a decline in the population heading toward extinction, but then you have a chance that maybe some members of the population will survive, but then what you do is you have a narrowing of the population genes. Okay, you have a narrowing of different alleles that the population can have. So that's what you call a bottleneck, because of course a bottle has a narrow neck, like a wine bottle, right? So the ones that survive then go on to make new individuals of the same species, but if they do not recover, the species goes to extinction. Okay? So those are the three different kinds of genetic drift: the founder effect, random chance, and the bottleneck effect. Okay? Now, rates of evolution. So Darwin believed that evolution was a very slow and gradual process, but maybe other people since then have thought of what they call the punctuated equilibrium model. So punctuated equilibrium may be a better model, okay, of how evolution really happens. Okay, so in gradualism, you have species actually branching off and gradually becoming other species slowly over long periods of time. You know, like the building of a coral atoll island, a volcanic island, and then the decomposition of the island into a coral atoll. You very slow, gradual changes, making up for large changes in the long run. Okay, so gradualism is that you have slow, gradual changes over very long periods of time. Punctuated equilibrium actually says that maybe you have major spurts of evolution happening in 100,000 years or less, leading to new species. Then you have long periods where there's no change or very little change. Then you have a spurt again, and then long periods of little change. Okay, so the punctuated equilibrium model is actually based on the fossil record, which has large gaps in it. Okay, so under climate change, like what's going on now, human-caused climate change, or other selection pressures, okay, you could have new species arising in less than 100 generations, okay, less than 100,000 years, okay? Now we also have macroevolution, how species actually evolve, okay? So in order for this to happen, you need some kind of isolation barrier that divides members of a group, okay? So if a barrier divides a population, the two populations may become different from each other, okay? And the isolation barriers can be pre-zygotic or before fertilization, or post-zygotic or after fertilization happens, okay, in sexual reproduction, okay? So pre-zygotic barriers include geographical isolation. So geographical isolation is like when you have intervening lake between two populations, or you have a mountain between two populations, and it prevents gene flow between populations. Okay, and mutations that might actually appear in one might not appear in the other. So for geographical isolation, we have an example of the redbuds or Circes species. Okay, so you have the eastern redbud that is usually east of the Mississippi River, and you have the western redbud. Okay, the eastern and the western redbud. And the western redbud lives in California, in Utah, in Nevada, and Arizona along riverbanks. Okay, so each of these two different species, well, they look kind of similar, but they're so adapted to the habitats that they live in that if you transplant one into the habitat of the other, they'll tend to die. Okay, so there are possibly two species, the eastern redbud and the western redbud might have evolved from one. That's called geographical isolation. Okay. Another kind of isolation barrier that's prezygotic is ecological isolation. Okay, so Dutchman's breeches, okay, actually looks sort of like baggy pants. Okay, so Dutchman's breeches actually is an early spring flowering plant. Okay, and it grows together in the same places as squirrel corn. So they're the same genus, but they're different species. Okay. So they go together, but their flowering times are different, so they can't breed together. That's an example of ecological isolation. Okay, some people also call it temporal isolation when flowering times are different. So that prevents them from breeding with each other. That actually keeps the Dutchman's breeches and the squirrel corn populations separate from each other, so they're different species. Okay. Then we have mechanical isolation, such as in catasetum orchids. This is actually a look at them. Remember we talked about catasetums before? These are unisexual orchid. They have unisexual orchid flowers, which is unusual. Most orchid flowers are bisexual. Okay, so let's see. We can actually have artificial hybrids that are easy to make by humans. Okay, between 4 different species, but in nature, no natural hybrids are known. That is, among the 4 species, there's no crossing between them, even though all of them are pollinated by the same bee, okay, same species of bee. What happens is that in the flower, in the male structure, here's the trigger hairs, the trigger points here that you hit the trigger and it'll actually fling polonium on an orchid. Okay, pollinia. So the pollen are in packets called pollinia, okay? And the pollinia are actually packaged together, and you have species number 1. So the pollinia actually attaches to the one insect's head, and for species number 1. For species number 2, it attaches to the back of the insect. For species number 3, it attaches to the abdomen of the insect. And for species number 4, it attaches to the left front leg. Okay, so the pollinia needs to be first attached to the insect and then actually inserted into the stigmatic concavity on the flower. It's like a concave thing, so it has to be like a fit, like this, almost like a lock and key. So if the insect has this on its head, it can only put the pollinia in a certain flower in a certain way. So that actually allows for a mechanical isolation between one species and another, catasetum orchids. Okay? Okay, now so prezygotic barriers actually are more significant than postzygotic barriers. Okay? Because at fertilization point, it's important for and wasteful for the mother plant to give energy for making hybrid seeds if they won't grow or they won't be fertile. So the postzygotic barriers to hybridization include the death of the zygote, that the zygote is conceived, or actually, the sperm and egg come together and fuse, but then the zygote dies. Okay. You can also have hybrid sterility when the hybrid plant actually is sterile, like it'll actually germinate from seed, it'll grow, and it'll bloom, but then it won't actually make more seed itself or fruits. Okay. And you also have hybrid low fitness, where the hybrid dies before it can reproduce. Okay, so these are the post-zygotic types of barriers to hybridization. Okay. Now the role of polyploidy. Okay, almost all angiosperms are polyploids. Okay, this is something we talked about before. Okay, now polyploids are much more common in plants than they are in animals. Okay, because for one thing, animals cannot tolerate the imbalances that come from doubling chromosomes. Okay, for a second thing, the reproduction systems in animals are disrupted by polyploidization. Okay, and thirdly, polyploid often occurs in hybrids between different species, which is common in plants but uncommon in animals. Okay, so diploids are generally, you can have a diploid hybrid. Okay, you have one set of chromosomes from mom, one set from dad. Okay, so this is a hybrid between two different species, or two different species plants, or even in two different genera. Okay, you might have different chromosome numbers. Okay, so if the offspring undergoes meiosis, the chromosomes may not pair during prophase 1 or meiosis 1. Okay, so what you have is a plant that's sterile, which is an evolutionary dead end, because fitness has to do with reproduction. So if you're an organism and you reproduce yourself, you are actually evolutionarily fit. Okay. No, but if the chromosome number doubles in the hybrid, you have two sets from mom and two sets from dad. Okay? Then they can pair, and then you can have a fertile hybrid. Okay, they can pair with each other. Okay? So now polyploid that comes from a cross between different species is called an allopolyploid. Okay, and a polyploid that comes from one species is called an autopolyploid. So it can happen in both cases, between different species hybridizing. You can have polyploidization, and you have one species that actually breed, and then you can actually have autopolyploidization. Okay. Now our daily bread. Okay, so we have cereal crops such as wheat, that is the foundation for agriculture for the last 10,000 years. Remember that cereal crops were among the first crops domesticated by humans in the Fertile Crescent. You remember, in western Iran? Okay, so wheat actually came from a wild allotetraploid. Okay, allotetraploid. Okay, from different species. It was more vigorous, higher yielding, and more broadly adapted than the wild diploid parents. Okay. Then through breeding, we got emmer wheat, which is used to make pasta. You have a higher yielding kind of wheat. Then you had that tetraploid wheat, and they crossed it with a wild relative, a diploid wild relative. So you had an allohexaploid with 6 sets of chromosomes. Okay, so that gave rise to modern bread wheats. Okay, you have a diploid wheat with 14 chromosomes, the wild ancestor with other chromosomes. So what you ended up with is you had the polyploidy occurring in the hybrid. So what you ended up with was a tetraploid wheat that had 4 sets of chromosomes. Okay. And this led to emmer wheat and durum wheat. Okay, then you had another cross with another species. Okay, then you ended up with a hexaploid wheat with 42 chromosomes. Okay, so you had 6 sets of chromosomes. So this led to what they call spelt wheat, bread wheats, and what they call club wheat. Okay. So the third genome had genes for certain proteins. Okay, that led to bread quality, good bread quality. Okay, allowed for a broader range for the plant to grow in. Okay. And now we have hexaploids making up 90% of wheat production. Okay, all over the world, hexaploids. And a problem, though, is a narrowing genetic base of cereals. Okay, we have only a few cultivars. So what they want to do, of course, is they want to broaden the gene pool. Okay, so what they'll do is they'll cross it with its wild relatives. Okay, so you can cross wild relatives and then double the chromosomes and make new polyploids, right? You can cross wheat with wild relatives. Okay, so this is actually a response for human population growth and also for global climate change. It's critical to actually assess genetic diversity because we want genetic diversity. We don't want another, bunch of corn dying or a bunch of potatoes dying, making starvation the norm or something that we have to go through. Okay, so what they want to do is they want to breed high quality cereal crops because I guess we're not really going to give up on cereals, even though some people are allergic to the protein in wheat, some people are allergic to it. Okay, now apple mixes. Okay, is actually when sterile hybrids may reproduce asexually. What they'll do is they'll produce seed without the benefit of fertilization. Okay, so what happens is a cell in the ovule undergoes mitosis to produce an embryo. Okay, so an example of a plant that does this is dandelions, one of the most successful plants in the world. Okay, they can actually reproduce apomictically as well as sexually. They can do both. Okay. Now, Darwin's theory of evolution was a great controversy during his lifetime. Okay. Although Darwin believed in a divine creator, okay, he believed that this divine creator actually allowed for changes in living things, gradual changes in living things, which would lead to things that are living that exist now, that it allowed for change. Okay, because remember that Darwin actually studied to be a minister. He studied theology, right? So until the end of his life, he believed in a creator. Okay. And also the thing is that another reason why it was a great controversy in his life was, of course, of his contemporaries, the people that lived during his times. Many of them, and probably most of them, believed in a literal interpretation or the biblical interpretation of how things came about, or in creation. That many people believe that in special creation, that you have individual animals and plants being created one by one in 6 days. Okay. And that only happened a few thousand years ago, okay? But what happened too is that science deals with facts and evidence that can be measured or tested, okay? That these are facts and evidence that are physical, that you can smell it, you can see it, you can hear it, you can taste it, you can touch it, okay? Empirical evidence, all right? So belief or faith is outside of the realm of science to disprove or to prove, okay? So one of the things to remember too is that people are allowed their beliefs. They should be allowed what they believe in, okay? Anyway, to sum up what we just did, okay, last time and this time we talked about evolution. We talked about the first, second, and third revolution of evolution. We talked about Charles Darwin, the author of the theory of evolution. We talked about the evidence for evolution. We talked about microevolution, macroevolution, and finally we talked about polyploidy. Okay? So now we're going to actually switch topics, okay, and get into plant names and classification. Okay? So all organisms on Earth that we know of are given one two-word scientific name, okay? There's only one correct name per species, okay? So for each species, you only have one two-part scientific name. Now you have many species that have many common names. A good example locally is Scaevola sericea. That's a scientific name for an alpaca, Okay, so it's actually, in English, it's called skivala or half flower, two different names in English. In Hawaiian, it's known as naupaka kahakai, naupaka kai, huahekili. On Ni'ihau, it's known as aupaka. Okay, so this is a plant that is actually indigenous to Hawaii, so it occurs in other places as well. Like in the Society Islands, it's known as ngau. In the Cook Islands, it's known as ngahupa. In Tonga, it's known as toi toi. These are all pictures of Skirilla sericea. In Samoa, it's known as viveda. In Guam, it's known as nanasu. Okay? So you can have that where you can have one species and many common names. Or you could have one common name for different species. Okay? So an example of that is mock orange. Here's Philadelphus. Okay? So you have Philadelphus species in the temperate areas. We have Maclura pomifera. Okay? Also known as Osage orange, but it's also called mock orange. See how different these plants are? And you have Murraya paniculata, which we know in Hawaii as mock orange. It's used oftentimes for hedges. Okay? So we have three different plants from three different families. They're not that closely related to each other, although all are dicots, and they're all known as mock orange. So there can be confusion if you don't use the scientific name. Okay? Now, binomial nomenclature. Theophrastus in the 4th century, okay, was the first person to organize and classify plants. He was a brilliant student of Plato and Aristotle, okay, and he's now called the father of botany, okay. He actually classified 500 plants, okay, as trees or shrubs or herbs, okay. Trees, of course, are woody plants that have branches more than halfway up the stem. Shrubs are woody plants that have branches from lower than halfway down the stem. And herbs are herbaceous plants with soft growth. Okay? So he actually divided 500 plants into trees, shrubs, and herbs. And he also distinguished between plants due to characteristics of the leaf. Okay? So the system that he used was used for hundreds of years. Okay, hundreds of years, right? Now, about the 13th century, okay, there was finally a distinction between the flowering plants, dicots and monocots, the dicotyledonnae and the monocotyledonnae, and that was on the basis of stem structure. So here's a monocot with one cotyledon, and here's a dicot with two cotyledons, and here you can see the young true leaves coming out at the apical meristem. Okay? So by the 18th century, people were also using fruit and flower characteristics too. Okay? So by this time, European institutions such as universities, such as herbariums, they bulged with thousands of plants brought back from around the world. Because remember, in the 1400s, 1500s, 1600s, the Europeans went on their voyages of discovery, their voyages of discovery. Okay, so there was growing confusion over scientific and common names. Okay, they used Latin phrase names. Okay, so plants were grouped into genera. Okay, genera, singular is genus. Okay, so this is for plants and animals both. Okay, with the first word in this Latin phrase name being the genus name. Okay. For example, spearmint. Okay, this was the name, a polynomial. Okay, Mentha floribus spicata folius oblongis serratus. Okay, what does that mean? It means mentha with flowers in a spike, leaves oblong, sawtooth. So this whole polynomial, or many name, actually stood for the spearmint. Okay, so you could imagine it was pretty cumbersome. Okay? So Linnaeus actually put some of this confusion to rest. His name was Carl von Linné, right? What he did that was so important was he improved the way that organisms were named, okay, and how they were classified. So we still use his system today, okay, throughout the world. Okay, he was a professor of botany and medicine in Uppsala. Brilliant man. He inspired large numbers of students. Okay, 23 of them became professors. Okay, he led large field trips into the countryside to prospect for plants, along with like a band playing, you know, like a musical band playing to them marching into the countryside. Okay, very popular professor, and he was quite a character. Okay, and in 1753 he published the book Species Plantarum. Okay, and it was the first book used by plant taxonomists, people who study plant classification, plant naming, and also to assign names to the plants. Okay, so he placed one or more species in a genus by how they were related. Okay, and he limited the Latin phrase name to 12 words. Okay, and in the margin of his book, Species Plantarum, he wrote one word, you know, that was like a descriptor. Okay, so he was the first to assign names, okay, in this form that we know it now. So that long polynomial we were talking about a little earlier is now known as Mentha spicata. So this spicata was a word that he used in the margin. So that, and this L stands for Linnaeus, because he was the one who actually put it in this classification. Okay, so he actually named the spearmint Mentha spicata. So he and other people eventually replaced the phrase names with these abbreviated names. Okay, so it's a two-part name, a binomial. Okay, so it follows what they call the binomial system of nomenclature. So all species today are named with this system. Okay, and generally this system includes the author for the name. And in this case, it was Linnaeus. Okay. Now, what he did too is he grouped plants by the number of stamens. He called it his sexual system. Okay. He grouped plants into 24 classes. Okay. But this was an artificial system. So he'd actually group all plants that had 5 stamens into one class. He grouped another group by 6 stamens into another class. So this was an artificial system that was not really reflect natural groupings. Okay. Now a plant that's new to science, somebody who discovers a new plant has to, first of all, publish a Latin description in a journal or other publication that happens to be in circulation, okay, and is available to the public, okay. They also must deposit something called an annotated herbarium specimen in a place called an herbarium. It's called a typed specimen. So this is an herbarium specimen. It's actually a dried plant mounted on an herbarium sheet, which is acid-free paper. Okay, so what these can actually allow this herbarium specimen to exist for centuries. There are actually some in the Bishop Museum herbarium at the Bishop Museum that actually came from Cook's third voyage when Hawaii was first discovered by the West in 1778. Okay, so there's some specimens that date all the way back to 1778. There are some herbarium specimens in some European institutions that date back to the 1500s. Okay, so they have to be put under controlled conditions, but they can actually last. Okay, now the first classification was into 2 kingdoms. You had the plant kingdom and the animal kingdom. Okay, so this is a system that works for the more complex plants and the more complex animals. Okay, but for other organisms it doesn't work as well. Okay, for example, you have an organism known as Euglena, or the euglenoids. Okay, there are 300 species of euglenoids. These are microscopic single-cell organisms. Okay, so let's see. So the differences between plants and animals. Most people would say that plants are green, right? plants don't move and plants don't eat. Okay? Whereas you would say that animals actually do move, animals are not green, and animals eat plants or other animals. Okay, but here you have an organism that ingests food through a gullet. Okay? It moves with the means of a flagellum. Okay? It also has chloroplasts. Okay? Many of them have chloroplasts anyway, so sometimes they can do photosynthesis. Okay? So there are two people, Hogg and Hickel, propose a third kingdom in the 1860s, which is called the Protoctista. Okay, so these are animals that do not develop complex tissues, and the fungi and the algae and the bacteria were placed in that kingdom. Okay. So by 1969, a scientist named Whitaker proposed a 5-kingdom system. Okay, so this was in place for a while. Okay, so there are 3 kingdoms of more complex organisms that were based on mode of nutrition, such as doing photosynthesis, the plantae, okay, ingesting food through a digestive tract or the animalia, okay, or the absorption of food in solution or the fungi. Okay, then you had 2 kingdoms of protistans that were different in terms of cellular structure. You had the prokaryotes that were put in the monera, and that you had the eukaryotic protists that were put in the protista. Okay? So you have the monera, the protista, the plantae, the fungi, and the animalia in Whitaker's 5-kingdom system. Okay, now in the 1980s, Carl Woese thought that Monera, the kingdom Monera, should be divided into 2 groups. Okay, and these were the archaea and the bacteria. Here they're shown as archaebacteria and eubacteria, but these are the archaea and the bacteria. Okay, so this splitting of the bacteria kingdom or the Monera kingdom into 2 different kingdoms actually started to be accepted in the 1990s and is now pretty much universally recognized. But now we have 3 domains. Okay? You have the bacteria, the archaea, and the eukarya. Okay, you have differences in RNA between these groups. Okay, you have differences in cell wall content between the bacteria and the archaea. Okay, other features that make them different from each other or distinct from each other. Okay, so most known prokaryotes are in the group bacteria. The other prokaryotes, which tend to be extremophiles, they tend to live in extreme environments, are the archaeans, are the archaea. Okay, now all organisms that have eukaryotic cells, that have a membrane-bound nucleus, membrane-bound organelles, okay, they're all in the eukarya. Okay, so they include the plants, the fungi, the animals, other groups that are in the protista, that were in the protista kingdom. Okay, so you have the bacteria in archaea and eukaryote domains. Okay, now if we were to do a complete classification of onion, we would put it in the domain eukarya, the kingdom plantae, the division Magnolia phyta, the class Liliopsida, the order Liliales, the family Liliaceae, the genus Allium, and the species Allium cepa L for Linnaeus. So you'll need to know this classification system. Okay, so I can give you a mnemonic, another way to easily remember these. Okay, so the beginning of each classification or each taxon, D, K, D, C, O, F, G, S. Think of King David hiding, okay? Dear King David, come out for goodness sake, okay? Dear King David, come out for goodness sake, okay? So you'll need to know all these levels of classification, okay, for purposes of your exam. Okay? Now, taxonomists are specialists who identify, name, and classify organisms. Okay? So there are taxonomists who do these. That's their sort of specialty. Okay? Now, taxonomists who also count DNA sequences and evolutionary processes to sort out these relationships are called systematists. Okay? Actually involve studying systems. Okay? Now we have different kinds of species concepts. Okay? The species are usually the basic units of classification, and there are at least 22 different ways of classifying species. Okay? First of all, we have the morphological species concept. It's kind of based on growth forms or morphology, and it relies heavily on herbarium specimens. Okay? So this can be very subjective. So traits may be unclear or inconsistently used depending on the taxonomist. Okay, so that's a drawback for the morphological species concept. Okay. Now the interbreeding species concept is one that many textbooks focus on. It's based on breeding behavior, what breeds with what. So a species in a population is able to interbreed and is reproductively isolated from other groups. Okay? Now the thing about that is that plants have different degrees of crossability, and it's hard to predict which groups can cross and which groups can't in nature. Okay? So they usually do greenhouse studies where you can cross things, but this may not be what happens in nature either. Okay? So you have orchids that can intercross species. You even have orchids that can intercross at the genus level. Okay, but in the wild, maybe they don't breed. Okay. We have also the ecological species concept, which is based on related individuals occupying a particular ecological niche. So we then assume that the evolution of species is governed by ecological differences. So you might have an oak that's able to breed together with another oak. Okay, they might grow near each other, but you can actually have them in different kinds of ecological niches. Like you might have oaks that grow in swampy wetlands, or you could have those that grow in heavily wooded areas. So you have completely different ecological niches, so there would be different species. Okay. We have the phylogenetic species concept, where phylogenetic history is determined, okay, by species boundaries. Okay. So phylogenetics is study of evolutionary similarities, okay, among groups. So individuals that have common evolutionary background are considered a species. Then we have the eclectic species concept that actually thinks that all criteria must be considered. Okay, we have the morphological species concept, the geographical species concept, the biological species concept, ecological criteria. They all must be considered. Then lastly, we have the nominalist species concept, okay, where species themselves do not exist. Okay, they suggest that only individual organisms are real. Okay, any determination of higher groupings, such as genera, families, and species, results in an artificial classification. So in this nominalistic species concept, the unit of importance is the local interbreeding population. Okay? Okay, so let's review what we talked about just now. We talked about common names versus scientific names. We talked about classification, and we asked the question and tried to answer, what is a species? Okay? Yes, anyway, so that was kind of a lot of things to cover. Okay, be sure to contact me if you have any questions, please. Yeah, really feel free to contact me, okay, by voicemail, by email, come in person and see me, okay, if you have any trouble or any questions, all right? So until next time, we say aloha to you, and we'll see you next time. Bye-bye.